All right, so you watched the videos, yes, before you're coming to this video. So, transcription and translation. So, again, a quick review. The word transcription. Think of like transcribing your notes. DNA is in the nucleus, it cannot leave the nucleus. And a section of the DNA is the gene. So, think of it as a recipe. Again, grandma won't let you take the recipe home, but she'll let you make a copy. So DNA can make a copy of RNA, make a copy, and we call it RNA. mRNA for messenger RNA. That's the quote copy of the gene. It leaves the nucleus through the nuclear pores, goes into the cytoplasm, and then translation happens where mRNA, ribosome, tRNA, the T stands for transfer, tRNA, amino acids, bring them together, and we build a protein, and this is the end product. So you can think of transcription, DNA to RNA, that's transcription. Then RNA to protein, that's translation. So now to talk about exactly how that happens. You guys saw the videos, yes? Transcription happens with the help of an enzyme. The enzyme's RNA polymerase. And again, a picture is worth 1,000 words. The videos are worth more, okay? So the RNA polymerase is an enzyme. It opens up the DNA, unzips it. So here's the DNA, then it opens it up, brings in complementary bases only to one side. And so ATCG, and RNA has the ACG, but instead of T, RNA has U. And if we look at RNA, it's single-stranded. It has the base uracil instead of thymine. RNA leaves, and again here, every 3 bases is called a codon, codes for one amino acid. There's the tRNA transfer RNA, brings in the right amino acid. If it has the right complementary bases, brings in the right amino acid. We build the chain, and then there's what's called a stop codon, and we release it, and we have the amino acids. Okay, the protein. All right, so let's continue. So transcription, copy of gene is made. Translation, it's used to make a polypeptide. Poly means many. Peptide bonds are the bonds between the amino acids. So let's look. So here is the picture of transcription. So here's DNA with the help of the enzyme, RNA polymerase, unzips the protein, unzips the DNA, brings in complementary bases, and here's the mRNA, the messenger RNA. When it's done, the DNA zips back up, okay? So here's a picture of it. It says transcription is the first in a 2-step process producing proteins, okay? In transcription, a single copy of one specific gene is made to form mRNA. All right. Translation, the mRNA is used to make molecules, specifically their proteins. So the things that need to be there have to be amino acids. Remember, amino acids are building blocks for proteins. There have to be ribosomes, and there's another RNA molecule called transfer RNA. By the way, DNA makes transfer RNA. DNA makes messenger RNA. DNA makes transfer RNA. So imagine that messenger RNA, that copy of the recipe, goes into the cytoplasm, gets together with the ribosome. Every 3 bases is called a codon. And there are transfer RNAs floating around. If they have the right complementary bases, so A, U, because this is an RNA, CG, GC has the right anticodon, brings in the right amino acid. This is called a codon chart. We're going to do this in lab where you're going to see it. Let's say the mRNA molecule, so let's say ACG, and it's like which amino acid would come in. If you look, there's right? Threonine. So if we look, go back. ACG. The first base was A, so we would go here, A, so we're here. The next base was C, there's C. So A, C, and the last base was G, G would go across. So for ACG, bring in the amino acid threonine, and that's what they show, ACG, threonine. Let's look at another one, and you guys can think about it. What if it's CAA? CAA. So on the messenger RNA, if it's CAA, what would be the amino acid that comes in? Okay, C, first position is C. So there's C, so we're going to be in here, C. If it's A, there's A, second position. So C, A, and then if it's A, we go across. CAA, we bring in the amino acid glutamine. Now, people, this is not like pretend makeup stuff for this class. This is the real deal. It's taken years to figure out the codons and the codon chart and what brings in the amino acid. And people, this is universal. Universal. By the way, there's always a start codon. So if you look, start codon is AUG, brings in methionine. And there are always stop codons. So UAA, UAG, UGA, those are the stop codons. So here is showing translation, right? We're here, we're in the cytoplasm. Here's the mRNA, here's the tRNAs, bringing in the amino acids. And you looked at the videos, right? You looked at the videos. The amino acids will link. That tRNA takes off, brings in another amino acid, maybe comes in again, blah, blah, blah. So the process has a start, right? There's initiation, we start. There's elongation, putting on the amino acids. And then there's termination when we hit a stop codon and we stop. And when it stops, they release. And then now this chain of amino acids is the newly built protein. So translation, the second step in a two-step process in which DNA directs the synthesis of proteins. In translation, the information from a gene that has been encoded in the nucleotide sequence is read. Ingredients present this in the cell's cytoplasm are used to produce a protein. So gene regulation, genes can be turned on, they can be turned off, they can be blocked. And again, this is a huge, subgroup of biology. There are tools, it says powerful tool used to monitor expression levels of thousands of genes, looking at gene expression. So for example, are some people more prone to be depressed than others, and does it have to do with their genes? We see that there is a truth to that, okay? So again, there's that nature versus nurture. The nature is our DNA. So it says lonely, isolated individuals have very different patterns of immune system gene expression. So we can have positive control, we can have negative control, we can have things that block the polymerase from attaching and doing its job. We have regulatory genes. So promoter, operator, regulatory gene that can influence, right, if things are turned on or not. So eukaryotes, also we understand this process rather well. Well, I'm sure there's a lot more to understand. Now, how about if there's damage to the DNA? What is that? Damage is called a mutation, right? So damage to the genetic code has a variety of causes and effects. So here we see some fruits and vegetables, and we see some abnormalities, and they're caused by mutations. So the word mutation, okay, it's a change in the DNA. Sometimes it's harmful. Most of the time we think of harmful effects, but sometimes it can be beneficial, and sometimes it has no effect. Oh. So what causes mutations and what are its effects? Alteration in the sequence of bases can lead to changes in the structure function of the proteins, can have a range of effects. For example, breast cancer in humans, a lot of cancers have been linked to mutations. If you've heard of BRCA1, BRCA2, these are two human genes. It says more than 200 different changes in the DNA sequence, and they can increase the development of breast cancer. So that nature-nurture thing. So some people inherit those genes, and if they inherit them, the likelihood of getting cancer is much higher. So for a certain type of cancer, like breast cancer. So for example, if you ever hear, that breast cancer is inherited or it's in families, that's true. All right, more likely to get it. So if a mom or a grandma or an aunt has breast cancer and another aunt, then the likelihood of inheriting those mutated genes that can become, cancerous genes are higher for those people. So they have to be careful and do self-examinations and get checked and all of that, and be very aggressive in how they treat it, if there are new lumps or bumps and so on. Showing a mutation in a fruit fly. Mutations in sex cells versus non-sex cells. You can imagine mutations in sex cells are pretty dramatic because they're going to affect your progeny. They're going to affect your babies, which get passed on. It's the ones that happen in sex cells. So types of mutations. If this is a normal DNA, right, and then making mRNA, making a protein. So they're showing, here are the amino acids. It's purple, green, yellow, right? Sometimes a mutation could be a base substitution. Like, again, something happens and the base, let's pretend it was an A, now it changes to something else. That's a base substitution. Sometimes a mutation can be an insertion, meaning you bring in a new base, that's a mutation. And sometimes a mutation can be a deletion, we take out a base. Those are just some examples of mutation. So base substitution, insertion, deletion. And when that happens, it would change the amino acid sequence. And if we change the amino acid sequence, now we have a new protein. There's relocation, sometimes things, duplication, sometimes things can change. What are mutagens, okay? Things that cause mutations. There are things in our environment that can cause mutations. Smoking, definitely. Smoking has, I forget how many carcinogens, those are cancer-causing chemicals. Smoking has that, right? Lung cancer. If you think of number one killer in the United States, is cardiovascular disease, so heart, cardiovascular problems. Number two is cancer. And out of cancer, number two killer in the United States is cancer. And out of cancer, the number one is lung cancer. And if we look at lung cancer, the biggest contributor to that is smoking, okay? Smoking, by the way, vaping, chewing tobacco, those are all bad things because the chemicals in there cause mutations, okay? This would be a result of the sun, so sunburn, right, radiation. This, x-rays, right, they can cause mutations. That's why they, if you go to the dentist and they put that big lead jacket on you, they're trying to protect you, okay? And supposedly, well, not supposedly, the new x-rays use a lot less radiation, but still you want to be careful. You don't want to do it often or have too many done. So why do dentists put on a heavy apron over you? Yeah, that's why. Dangerous, okay, yeah, okay, well, I'm gonna stay away from that. So mutations, alterations, single base, or changes in large segments of DNA, they are rare, but when they do occur, they may disrupt normal functioning. Extremely rarely, mutations may have a beneficial effect. So mutations can be harmful, can be neutral, meaning there's a mutation, but it still codes for the same amino acid. And if it still codes for the same amino acid, there's no mutation, no change in the protein. Or rarely it could be beneficial. Faulty genes, coding, right? Can some people be affected by alcohol more severely than others? The answer is yes. We actually have figured out what gene, there's a mutation where the person can't process alcohol that well, so the alcohol really builds up in their system and they get drunk faster. That's real. Yes, with certain populations, we see that. All right, so we're going to go ahead and stop here for right now.